Good morning and welcome to this bio. Now we are going to discuss about Ramachandran plot. It was proposed by G. and Ramachandran to study the structure of proteins. Peptide bond formed in between amino and carboxyl group of amino or different amino acids that form a polypeptide. Peptide bond formation is thermodynamically not favorable. That is, it is known as an endergonic reaction or endothermic reaction that need some energy in the form of ATP to form a peptide bond. Partial double bond character of the peptide bond has partial double bond character that keep the structure rigid and planar configuration. The bond, that is peptide, polypeptide can be shown NCC, NCC like that. So the bond formed between C alpha and the CO is shown by psi. The bond formed in between C alpha and CO, CC psi. The bond formed in between C1 and H is shown by omega, which is called peptide bond or rigid peptide bond. The bond formed in between NH and C alpha is shown by phi, it is phi. NH and C alpha is phi. So the partial double bond character of peptide bond can be seen in this figure. So it is one of the another canonical forms that showing partial double bond character between in the peptide bond. So normally, if you see the bond length in normal cases in between C and N is 1.45 angstroms, and the bond length in between C and N in peptide bond, that is C1 and H, is 1.33 angstroms. So this is less than this one. It is showing that it must have some partial double bond character. In fact, it is one of the evidences showing that peptide bond has partial double bond character. Then psi, phi, and omega is called torsional angles. Shape of peptide chain defined by three consecutive torsional angles. These are phi, psi, and omega. For phi and psi, any values in between minus 180 and plus 180 they are possible. So, but within this range, many values inhibited by steric hindrance. So, if you see the bond rotation and torsional angle, the bond formed between NH and C alpha is free to rotation. It is shown by phi. The bond formed between C alpha and CO, CC psi. So the bond formed between C alpha and CO is free to rotation. It is shown by psi, CC psi. So the bond formed between C1 and H is restricted rotation, having restricted rotation. It is shown by omega. Two configurations are possible for planar peptide bond, that is cis and trans. You can see in this case trans configuration showing CO and NH opposite to each other having 180 degrees of angle. So this is called trans configuration. Trans configuration is more stable than cis configuration. So Ramachandran worked on commutative values of pi and psi and plotted a 2D map called Ramachandran plot. So it was proposed to know the structure of dipeptide. So now it can be used for a polypeptide having any number of amino acids. So Ramachandran plot for L amino acids and D amino acids are diagonally opposite to each other, which we can get from these diagrams. It is for L amino acids, this is for D amino acids. If you see in this case, this structure can be divided into 4 parts. Part 1, 2, 3 and 4. It is also divided into 1, 2, 3 and 4. Horizontally we have shown pi values, and here in this case in y-axis we have shown psi values. So the same even here for the acids, here phi values, it is psi values. So pi values are in between minus 180, 0 degrees and plus 180, that is minus 180 to plus 180 degrees, and here these psi values also minus 180 to plus 180. So in part 1 we can see anti-parallel beta sheet, and the lower case with the showing parallel beta sheet. The next one here in this case in part 3 we can see right hand alpha. In part 2 we can see left hand alpha. This is for L amino acids. In the case of D amino acids, these values are diagonally opposite to each other. If you see anti-parallel beta sheet in the case here, in this case, but in this case it is for anti-parallel beta sheet. So this is for parallel beta sheet in the case of amino acids. So here in this case it is for parallel beta sheet. So we can say these two are diagonally opposite to each other, right? These two are diagonally opposite to each other. So here we are showing right hand alphabets. It is diagonally opposite to each other, means this one. This one is diagonally opposite to each other. This is for right hand alphabets. Then this is for left hand alphabets. It here in this case it is diagonally opposite. That means it is for left hand alphabets. Then glycine is the only amino acid having free rotation. If you see the structure of glycine, the alpha carbon having no bulky groups. It has only two hydrogen bonds or two hydrogen groups which are not so bulky. So it has free to rotation. It is free to rotate. As a result, it is mostly allowed in the Ramachandran plot. If you see, in the case of proline, has cyclic side chain, that is pyrimidine ring in its structure, which is mostly rigid. So we can say it is least allowed or most restricted in the Ramachandran plot. So the original Ramachandran plot is this one. So here we are showing pi values. It is for here we are showing psi values. It is also divided into 1, 2, 3, 4 parts. So here we are showing anti-parallel beta sheet. It is for the amino acids. Here we are showing parallel beta sheet. It is for left hand alphabets. Here we are showing proline. This area is for proline, and here we are showing right hand alphabets. If you see here, the dark one showing mostly allowed area. Dark one is most allowed area. The area showing with the dots is also allowed. The also is less allowed when compared to this one dark region. So the other region here in this part is also allowed, but the remaining part is not allowed for any structure of polypeptides. For Ramachandran plot, we consider only amino acid residues that are present on the backbone of the polypeptide and not on the side groups. Acetylated or methylated protein is allowed minimum in Ramachandran plot. Generally, we show Ramachandran plot for peptide having L amino acids, because if you see in the living organisms, L amino acids are most frequently available. So except in some cases like prokaryotic cell walls having D amino acids. So by using this Ramachandran plot, we can predict the structure of proteins but not the sequence of any amino acids. The structure of protein are pi values and psi values for some structures. So left-handed alpha is having pi value plus 157, psi value minus 47 plus 47, and right-hand alpha is having phi value minus 57 minus 47. So anti-parallel beta sheet minus 139 plus 135. So parallel beta sheet minus 190 plus 130. So collagen minus 51 plus 153. So proline having the minus 60 and plus 150. Thank you. Take a screenshot of it.